afternoon, everyone. I am Liao, and I'm from University of Florida, the Sunshine State. So this morning I wake up at 5 o'clock, and I want to wake up before the snow stops because I want to see it snowing, but apparently it's still snowing right now, so yeah, too early. And I'm a PhD student in the plant pathology department, and today I'm gonna share with you some of the research I've done in my master degree and a little bit of what I'm doing right now. And the title of it is Using Magnesium-Based Nanomaterial for Management of Bacterial Spot Disease of Tomato. And I talked to some of the people last night. They say they want to nod right after they hear that title. So I really try to be informative and entertaining today, but feel free to interrupt me if you have any questions. And if I can't answer you, I promise I will find somebody can, like my boss. Yeah. Okay. So let's get started. I think most of the people in this room know that tomato already joined the tomato production and know tomato is a really economical, valuable crop, not just in Florida, it's around U.S. And I'm just putting those big numbers there, showing how important it is. And Florida is the #1 fresh market tomato producer in the US. So based on that, we suffer a lot of bacterial disease because we're warm and wet in the environment. So today, I especially want to talk about is bacterial spot disease of tomato. And if you see the leaf over here, that you can see that spot, really circular spot, and with yellow margins around it, like a halo. And that is a really, really typical symptom for bacterial spot disease of tomato. And this kind of symptoms can appear on leaf, on stem, and on the fruits. So this is one of the field trials we do in balm that you can see that the lower canopy is all dry and distorted. So this disease can cause defoliation or fall of fruits, and so it's a really devastating disease, and it is caused by 4 distinct species of Pseudomonas, and the major yield loss can be up to 50% of the fresh market tomato. So that is a really devastating disease in Florida and around the world. Yeah. So now I want to show you the disease cycle of this pathogens, and because I drew this picture, so I'm really proud I'm gonna present it to you. And this picture is gonna be in the EDUS publication really soon from University of Florida, so if you want to check it out in the future. So you can see over here that the pathogen can survive in the plant debris or the seed, and you can continuously survive in the transplant. And after you put your transplant in the field, it is really, really hard to try to manage and control them. And because it can invade through natural opening into stomata or wounds, and because of the wind and rain, it can splash and infect the plants next to your diseased plants, and eventually the whole field might have bacterial spot disease of tomato. So there are several targets you can try to manage it. First, you can use the clean seeds are recommended. So if you listen to the section in the morning, Dr. Gugino introduced some of the methods of how to treat your seed. And unfortunately, there's no resistant cultivar of tomato at this point because the species and race shifted so rapidly, so it's really hard to maintain a resistant cultivar. And healthy transplants, but after it is in the field, after a disease in the field, basically there's no cure for it, so we can try to use chemical control or biocontrol. And the sanitation of the field is really important, so clean out the plant debris or clean out the weed or the volunteer tomatoes in your field after harvest. So that goes to the current IPM strategy, is including the cultural practice that I just mentioned that ideally can use pathogen-free seed and disease-free transplant and avoid overhead irrigation in the field, because just basically try to keep it dry, not too wet, because bacteria, they like the humidity. And a lot of growers use copper-based bactericide, including copper mangoseb, especially in Florida. They use that quite regularly to try to control bacterial spot disease of tomato. And here I'm showing you a piece of history of bacterial spot disease of tomato. This disease is first characterized in the 1940s. And in the 1950s, the antibiotic, including streptomycins, have been used quite often. But really sadly, unfortunately, in the 1960s, the streptomycin resistance had been developed. So after the resistance being developed, the copper-based bactericides having been used really regularly in the field, but not like really short after, the copper tolerant strains have been present in the field. And because bacteria, like this little comic over here, they like to transfer plasmid or horizontal gene transfer. So in the year of 2006 survey, out of the Xanthomum preferens, which is the dominant strain of causing bacterial spot disease of tomato in Florida, are copper tolerant. So this is not just a Florida problem, because a lot of states right now, they found copper tolerant strains of Xanthomonas. So this discovery caused the copper bactericide. They are unaffected. And there's some problem about the potential phytotoxicity due to heavy metal accumulation. So that makes finding an effective alternative chemical really important. So there are several alternatives, like either on the market commercially available or not commercially available, but they all have its pros and cons and inconsistencies. For example, the SAR inducer Atigar, that in some of the old research that said it might cause ear loss because you are triggering the plant defense and it might cause energy. But we didn't see that in the recent research. And for bacteriophages, it's sensitive to the environment, so it's like UV. And there's some of the biocontrol agents that it shows inconsistent from season to season. And there's some non-commercially available one that were being tested. And for like the biofilm inhibitor, you still need to combine with copper bactericide, so it's not really ideal. And for some of the nanomaterials that we tested, and it's have good antibacterial ability, but haven't been the copper-based nanomaterials that the Amanda Strayer introduced last year. That one is through the pipelines, but others it's not commercially available. So that leads to the question: so what is a nanometer? So if you have like the thread of hair over here, it's 100 micrometer, and our pathogen over here, the Xanthomonas preferens cell, is 1 micrometer. So it is 100 times smaller than the diameter of a thread of hair. And the material we are using now is around 100 nanometers. So it's 1,000 times smaller than the diameter of the thread of hair. So it's really small. So why we use nanomaterials? Because if you imagine the micron size of copper bactericide, like copper mangoseb or Coside 3000 as a coffee beans, if we reduce the size, like the coffee grinds, they have more surface area to contact with the pathogens and might release more ions, and the nanoparticles behave differently than their micron-sized counterpart. And there's some previous research that shows metallic oxide, including nano-magnesium oxide and nano-copper oxide, while you reduce the size, the bactericidal ability will increase. So that's why we are really interested in the metallic oxide nanomaterials, and we want to test them against bacterial spot disease of tomato. So our hypothesis over here is the metallic oxide nanomaterials will provide better antibacterial properties compared to commercialized micron-sized copper and more effectively control bacterial spot disease of tomato. So at first, we apply a bactericidal ability experiment. So we mixed the treatment separately, including the copper oxide nanoparticle, magnesium oxide nanoparticles, and to compare this with the copper bactericide CoSci 3000 and the untreated control. And we mix them with the pathogens into a test tube. And after 15 minutes, 1 hour, 4 hours, and 24 hours, we plate them and count the populations. And to give you a better idea of how a copper-tolerant strain of pathogen and sensitive strain of pathogens, they will react. Here I provide you a plate that these two plates, they contain 200 ppm of copper sulfate. But if you streak the copper-sensitive strains over here, it's really hard for them to grow. The copper will kill all of them. But for the copper-tolerant strains, they grow really, really happily on the copper sulfate plate. So that's why we are trying to use nanomaterials to kill them in this experiment. So this is the result of the bactericidal activity experiment. And the upper one is to test it against copper tolerance pathogen. And the lower one is to test it against copper sensitive pathogens. And the x-axis is the treatment that we've been using. It's 2 different concentrations of copper oxide particle, magnesium oxide nanoparticle, copper oxide nanoparticle, magnesium oxide nanoparticle, and the micron-sized copper bactericide CoSci 3000 and untreated. And the y-axis is the population. So we can see over here, after 4 hours, all the bacteria can be killed in the 100 ppm magnesium oxide nanoparticles. And after 24 hours, all the bacteria has been killed in the 1000 ppm copper oxide and magnesium oxide nanoparticles. So it shows it has bactericidal ability against the copper tolerant strain of the pathogens, and which the CoSci 3000, which is the copper bactericide, doesn't have that ability. And for the copper sensitive strain, as we predicted that the copper, all of them works pretty well toward the copper sensitive strain. So the magnesium oxide nanoparticles still can kill the copper sensitive strain, and in the meantime kill the copper tolerant strain. So we find it pretty promising. So that's why we put it into the greenhouse study that we can see in the next slide. Am I talking too fast? Okay. Okay, so in the greenhouse experiment, we basically, we use the plants that are around this big, and then we spray the treatments, including 4 different concentrations of the copper oxide nanoparticles, magnesium oxide nanoparticles, to compare with CoSci 3000, copper manganese, and the water control. So we spray them a day before we inoculate the plant to let it dry, and then we inoculate the plant with the copper tolerant strain of Xanthomonas perfringens, and then we bag them to create an optimum environment and unbag them, and then rate the disease severity, then calculate the average under disease progress curve. So we can see in this next slide that the upper graph is the disease severity, and the x-axis is 4 different concentrations of the copper oxide nanoparticles, magnesium oxide nanoparticles, and the copper bactericide CoSci 3000, and copper manganese grower standard, and the untreated control. And the y-axis is the average under disease progress curve. That you can see here for as low as 200 ppm of the copper oxide nanoparticle and magnesium oxide nanoparticle significantly reduce disease severity compared to the untreated control. Although the copper bactericide and grower standard they have the same level of disease severity reduction, but we are using a lower concentration. And for the 1,000 ppm of magnesium oxide nanoparticle actually have significantly reduced disease severity compared to the grower standard and COSI 3000. And this magnesium oxide nanoparticle doesn't cause any visual phytotoxicity in our greenhouse. And we repeated this experiment 3 times, and the trend is really similar. That's why we put it into the field study. So in the field experiment, we treat the block, we treat the field 1 time before we inoculate them with copper tolerant strain of Xanthomonas preferens. And then we do a completely randomized block, and we have 8 weeks of follow-up treatment with rating of disease severity. And then we calculate average under disease progress curve. So I'm showing you the first field trial that we've done, that the x-axis is 2 concentration of copper oxide nanoparticles and magnesium oxide nanoparticle to compare with COSI 3000 and copper mangazep and the untreated. And the y-axis is the AUDPC. And you can see over here that both concentration of the magnesium oxide nanoparticles significantly reduce disease severity compared to the untreated. And the grower standard copper mangazep and COSI 3000 doesn't significantly reduce disease severity in this field trial. So because of this interesting result, that we do another 2 field trials, one in Quincy, Florida, and one in Baum. And the same, although different field trials show different results, but there's one treatment is consistently shows disease reduction, disease severity reduction, which is the 200 ppm magnesium oxide nanoparticle. This one. And the COSI 3000 and copper mangazep doesn't significantly reduce disease severity in any of these trials. So we're safe to say here that magnesium oxide nanomaterials and 200 ppm consistently reduce disease severity in the field. So because we try to be, and the next field experiment will try to be more realistic, because the growers being spraying copper mangazep like twice a week. So if we figure if we spray copper mangazep twice a week, it might significantly reduce disease severity and might be more like the magnesium oxide nanoparticles. But we see over here, we compare the magnesium oxide nanomaterials with COSI 3000 and grower standard copper mangazep and the one treated. And in the intense application field trial, still only the magnesium oxide nanoparticles significantly reduce disease severity in the field. And there's another question come up, that is that only the smaller size of magnesium oxide that have that reduce disease severity? So that's why we compare the nano size over here, the 20 nanometers magnesium oxide particles, to compare it with the bigger micron size, which is this 2, with 2 different concentrations, and we still compare them with the copper bacterial side and the grower standard copper mangazep and the untreated. And you can see over here that only the smaller size, which is the nano size of magnesium oxide nanomaterials, show significant disease reduction compared to the untreated. So the next step, because we already see our material show significantly reduced disease severity, the next one is more like a safety experiment. So we sent our fruit from the field trial to CAES in Connecticut, and Dr. White helped us to homologize the fruit and then analyze what's the elements in the fruit. Because in the former study that it shows nanoparticle might affect the elemental intakes of the plants. or we are worried that a nanoform of copper oxide or magnesium oxide might accumulate on our fruit. So for the result that this experiment's being repeated twice, and for the result, there's no significant difference of magnesium or copper in nanoform in any of the treatment. And there's no significant difference for all of the elements that were being tested in the nanoparticle treated fruit to compare with the untreated. So at this point so this experiment is just a pilot one. If we want to do, we really want to go deep with this product, we might do more toxicology experiments. So there's another thing that the magnesium oxide nanoparticle that I've been using is non-formulated. So it tends to aggregate with each other. So sometimes they showed inconsistency in the greenhouse or in the field trial. So this graph I'm showing you is the x-axis is the size of the particle, and the y-axis is the intensity, which is the peak shows most of the particle is this size. So for the 1000 ppm of the magnesium oxide nanoparticle I'm using, it's not actually 20 nanometers that we think. So the higher concentration is, the size is bigger. That means that they tend to aggregate with each other, so it's not as practical as we predicted. So Dr. Sandra from UCF, our collaborator, and his group helped us to develop a formulated magnesium-based nanomaterials, which are easier to suspend, and so that's why we are testing it through the bactericidal, and we're testing with the bactericidal activity experiment that is the same experimental setting as the non-formulated one. Okay, and so we can still see here that after 15 minutes, all of the formulated magnesium-based nanomaterials kill all the bacteria in 15 minutes. And the same trend you can see against the copper-sensitive strains too. And in the greenhouse study that we compared our non-formulated nanomaterials with the formulated nanomaterials, and it's still, the formulated one still significantly reduced disease severity compared to the untreated control and the growth standard copper mangazette. So we found it really promising, so we're gonna put it in the field in spring this year to see how it works. So for conclusions, for bactericidal ability, magnesium oxide and copper oxide nanoparticles have better bactericidal activity compared to COSI 3000, which is a micron-sized copper bactericide. And for disease reductions, in the greenhouse, both magnesium oxide and copper oxide nanoparticles significantly reduce disease severity compared to copper mangazette. And in the field study, the magnesium oxide nanoparticles show significant disease reduction compared to the untreated control. And the smaller the particle is, the better and more significant the control, it shows disease reduction. And for the twice-a-week application, magnesium oxide nanoparticles show more significant disease reduction compared to copper mangazette. And there's no excessive metal accumulation in the fruit, and there's no phytotoxicity, and there's no significant yield reduction. And since we are using a really, really small scale for our field trial, it's really hard to reflect yield in our experiment. So our future work is we are going to use the formulated magnesium nanomaterials in the field to see how it works, and we're probably gonna combine it with other chemicals to see its potential of managing bacterial spot disease of tomato. So here I want to thank my committee members, Dr. Matthew Parrott and Dr. Jeff Jones, Dr. Josh Freeman, Gary Valette, and Neha Potnes. And I want to thank Dr. White and his group in Connecticut. And I want to thank Dr. Sandra and his group in UCF, and basically Dr. Jones' lab and Dr. Parrott's lab. And yeah, and I will happy to take questions if there's time. But one question.
We'll take one question. We got 2 minutes. Right up front.
So there's not a product on the market that we can buy today?
You missed my introduction, didn't you?
Sorry.
Yes. So,
I guess I did.
yes, sorry, yeah, my introduction was that this is forward-looking and hopeful. Mr. King, very hopeful, yes.
What was your delivery method for the magnesium oxide on the tomatoes? Were you using a conventional sprayer?
Yeah, we use a conventional sprayer. But for the bomb one, we use the press one. So we use different sprayer in the different field, these 2 field trial. But most of the trial we use conventional sprayer. Yeah.
She talked about compressed. That's a what's the gas they use pressurized?
Yeah, pressurized. Yeah.
All right, let's give her a hand.